this working? It's okay? Thank you for your kind words, David. Well, the title for my talk today is From the Earth to the Brain, and this is a very dangerous title, as you can guess, because I know that your expectations may be too high for me. But I will try to cope with this challenge. But let me first thank all these labs in Europe and America that includes many very good researchers working with us in the main topics I am going to present today. So please give me these 10 seconds for thank all these great people working with us. Well, for beginning my talk, I will use a very short video, and I will try to use this video for presenting my key message for the note today, okay?
We choose to go to the moon. We choose to go to the moon. We choose to go to the moon in this decade and do the other things, not because they are easy, but because they are hard.
That's beautiful. It's got to be one of the most proud moments of my life, I guarantee you.
Okay. Do you agree in that the moon is beautiful? It is. It is beautiful. And that's why we get there a long time ago. The same thing can be said about the brain. The brain is also beautiful. And my main message for today is that we must go there. So, are you going with me? If the answer is yes, then we can move ahead. And for moving ahead, I think it's a good idea to say something strong just at the beginning of my talk. And the thing I'm going to say now is that we, psychologists interested in individual differences, study many psychological traits. There are many interesting psychological traits, but there is one psychological trait that is the most important one. And this key psychological trait is intelligence. That's why I am telling you today that when we go from the earth to the brain, we must begin our trip studying intelligence. So using this representation, I am saying today that the first goal is analyzing this core psychological trait, and afterwards, perhaps we can take a look at the remaining traits. So let's begin with intelligence and see what happens when we analyze the brain, when we go to the brain and we focus on intelligence. For doing that, I will tell you some things today for arriving at the final message that all of you must keep studying what you are doing now and going with me for studying intelligence, the key psychological trait, by studying the brain. So that will be my final message for you today. And now, two basic questions for beginning my talk. The first one is that for doing this trip from the earth to the brain, for studying intelligence, we need, this will be not a surprise for you, we need genes. We need to take into account genes, but not by themselves, because genes build the brain from the very beginning. So I am showing now an example of the type of knowledge that will be interesting for us for analyzing the brain. This study was published recently, and it's based in, as you can see there, in 400 twins, and they arrive at a very interesting description of how different genes build different regions in the brain. So here you have the main findings in this study, and you can see 12 different regions that are supported by different genes. So these researchers were able to isolate 12 main regions, 12 regions that can be distinguished by the influence of different genes, but, and this is very important, these regions are related. So they are increasingly built in a pyramid, and perhaps this sounds familiar to you. I am right? So we have different areas in the brain, but we have one single brain. Okay? The second message, before moving ahead, is an interesting, very recent study done by my friend Sharif Karama in Canada. And this is a very interesting study because it shows that, in this case, cortical thickness is related with differences in intelligence when intelligence is measured at 11 years of age and 70 years of age. You see in this slide that in both images, the brain correlates of these IQ differences are almost the same for these two, the same subjects at different ages, and the correlates of variations in cortical thickness are almost the same. The interesting thing in this study is the answer to the question of what happens when you analyze the correlation between cortical thickness and IQ differences at 70, controlling for differences in IQ at 11. Okay? And can you guess what we obtain doing that? Nothing. So there is no correlation between differences in cortical thickness and differences in IQ when you control for the differences in IQ at 11. So there is a great stability in this brain index, and this is telling a very powerful story about how the brain is behind this core psychological trait. Okay? Well, a couple of years ago, Paul and I brought a very exciting chapter for this handbook, and we try to explain there the basics of the relationships between the psychometrics of intelligence and neuroimaging. And I think this is a very interesting chapter, and if you have some time, please take a look at this one, because I do not have enough time today for telling many interesting things. What we try to explain there, I will give a couple of ideas now, is that we can use neuroimaging for obtaining relevant information to improve our understanding of individual differences in intelligence. And we can do many interesting things, like analyzing voxels in the brain, analyzing cortical surface area. We can analyze cortical thickness. We can do many interesting things, but the important point is doing the right things, not many things. Just do the right thing. So we try to explain there how to use the most interesting techniques in the neuroimage field for increase our understanding of this key psychological trait. We can also combine these neuroimaging techniques with genetics, and there is a very interesting project, but maybe some of you know, and this project is the Enigma project. And in this project, people from different labs around the world are cooperating for combining genetic information and information about mainly, but not only, but mainly about brain structure. And they are trying to find keys about different things, not mainly about intelligence. And this is one thing I will discuss later. But they are doing that for trying to understand different things in psychopathology and so forth. But we can use also this powerful approach to increase our knowledge about intelligence. And this slide tells an interesting story that is neglected when, I will show you some examples about that is neglected when people doing neuroimaging becomes interested in intelligence. They forget about that. They forget that when you measure intelligence, you are measuring different things. And these different things are really interesting for obtaining knowledge about what is intelligence, about how differences in intelligence can be understood analyzing brain images. So here, we try to explain to neuroimage researchers why it's important to distinguish the different sources of variance when you are measuring intelligence and relating those differences, those measured differences, with different brain indexes. Okay? And here you have one example, one key example about what this means. Some years ago, we analyzed a sample with voxel-based morphometry, and we do this. We analyze different subtests from the Wechsler, and we compute the G-loadings of these different subtests. So you see there that block design shows a very high G-loading, almost 0.9, while the symbol shows a very low G-loading. We know what this means. This means that if all the subtests are measuring, say, 10 mental processes, then you can say that block design is tapping 9 of these mental processes, while the symbol is tapping just 2 mental processes. This is why the block design is showing a very high G loading. It's taking almost all the cake of the intelligent construct. Okay? So we found what you can see there just in 3 images. You see what happens with the symbol that shows a low G loading. You see what happens with picture completion that shows an intermediate loading in G, and what happens with block design. And it's easy to see here that more clusters in the brain are correlated with differences in performance with increased G loadings. So this was one of the interesting evidence proving that these things called G loadings that may sound like some psychometric, very complicated thing, it's reflected in the brain. Okay? So if you are studying the correlates of intelligence performance using different tests, it's important to know, for instance, the G loading of such test, because this will change the findings. You can see here that there is only a very small spot there. Is this the brain site for intelligence, or are these ones? So you will find many different things. We are using different measures for estimating differences in intelligence. So this is important. Some years later, we analyzed a representative sample of children and adolescents, and this is difficult. It's difficult to analyze a representative sample using neuroimaging techniques, and we try to answer this main question. Are cortical thickness correlates of cognitive performance still there when you control for the G component of these measures? Okay, is G the key for understanding the brain correlates of the intelligent construct? That was the main question. Okay, so we identify 3 primary abilities: school skills, verbal reasoning, and spatial reasoning, and the G factor. So what we did here first is analyzing the relationship between cortical thickness and these 3 primary factors, and you see that there are many interesting results in different areas of the brain. So there are brain correlates, physical correlates of these 3 primary abilities. What happens when you control for the G component of these 3 primary factors? What happens? This. So nothing. There is nothing when you control for the G component of these 3 primary abilities. So the main conclusion here was that the G component is key for understanding the brain correlates of the intelligence construct, using, and this is important, I think, a representative sample of the population. Okay? The same thing happens for the different subtests. When you analyze, when you move down in the pyramid and you take the different subtests and you remove the G component of these specific measures, nothing appears there. Okay? Well, it's highly likely that you know this model, the PFIT model, that tries to find the commonalities among different studies, analyzing the brain correlates of intelligence. This model suggests that there are many different sites in the brain that are important for understanding individual differences in intelligence. But not all the brain regions are relevant. There are some elements in the temporal lobe, in the occipital lobe, in the parietal lobe, and in the frontal lobe, but not all the sites in the brain are relevant for intelligence according to the PFIT model. The PFIT model underscores, highlights two main sites in the brain, and these two sites in the brain are located in the frontal and in the parietal lobes. So Jung and Heyer are telling that these two brain sites and their connections are especially important for intelligence. Well, is this true? It is interesting to try to find commonalities among different studies. It's necessary to find these commonalities, but is this true or not? Well, we tried to test that some years ago with a sample of 100 young adults. And we do that using the same model we just saw, trying to define 3 primary factors and a higher order factor defined in G. You see there that G and fluid intelligence are the same thing, and this is not a surprise. And we use voxel-based morphometry for finding the correlates of these different scores. We focus just on fluid intelligence, or G, and crystallized intelligence and spatial intelligence. And we found different results for fluid intelligence, or G, for crystallized intelligence, and for spatial intelligence. This was summarized in this picture, and the main message that we can obtain from this picture is that there are many, many places in the brain that are related with variations in fluid, crystallized, and spatial intelligence. You can find also some places in the brain that are relevant for fluid intelligence, crystallized intelligence, and spatial intelligence. And these common places for the 3 factors are also distributed across the brain. So are the frontal and parietal areas key for that? For understanding the intelligence construct? We don't think so. We think that there are many different places across the brain that are relevant for supporting differences in intelligence. And then we move one step further, and now I will present a couple of studies with people with brain lesions. And we did that because we were interested in moving one step further, and instead of just computing correlations, we were interested in talking about causality. Is there some brain areas that are causing differences in intelligent performance? And we did that using a very interesting approach that combines information from hundreds of patients with different lesions in different areas in the brain. And you see here a short example. And then you ask the next question. Is the lesion in this voxel affecting intelligent performance? And we do that for the different patients and across the brain. okay? And this is done using an approach that it's called voxel-based lesion symptom mapping. So we try to check point by point in the brain how the different lesions are affecting intelligent performance. So this is the main question. Which voxels preclude proper intellectual performance? And we found, I will skip that, we found that, as you see here, that mainly frontal and parietal areas of the left hemisphere are relevant for influencing intelligent performance. So this main finding supports the PFIT model with patients, and in the left side of the brain. But in addition to this main finding that was mainly derived from the G factor, from the G score, we ask the next question. Well, what happens if you compare what happens for G and what happens for the different subtests that you use for computing the G score? And try to compare the different findings, what is common to the different subtests, and is there something specific for the G factor with the remaining subtest? Okay? And we found a very interesting thing that is highlighted here in the frontal area, that it was special for G. It was only relevant for G, but not for the remaining subtests that we use for computing the G score. And this led to the conclusion that although we found a distributed processing across the brain, this processing is consistent with a control mechanism that we found in Brodmann area 10, in the lower part of the frontal brain. So there is something there, we thought, there is something there that is especially relevant for the main component of the intelligent construct. We did another study, again with patients, and we asked here if there is something common to two important factors. One is the general factor of intelligence, and the other one is executive function. And we did this study with almost 200 patients, okay? And we asked the same thing that I explained before. Is there some specific areas in the brain, boxes in the brain that are affecting intelligent performance and executive function? And the main finding here was that, again, mainly in the left part of the brain, lesions are influencing intelligent performance and executive function at the same time. So there is something relevant, according to these two studies, in the left side of the brain for general intelligence. But as you know, general intelligence is too general. So we move one step further and analyze what happens with a key component of the intelligent construct that is fluid intelligence, and look at what happens for these more specific facets or factors of intelligence that are beyond G. So this is a summary of the relevant areas that are common to different studies and different analyses that are mainly common to the PFIT model. But if you go to this lesion data and ask what happens, for instance, with block design, remember, a test with a high G loading, but that is not only G, you see that the main findings are on the right side of the brain for block design. And in the second lesion study, for the fluid intelligence factor, the main spot is on the right side again. So what is happening here? Is intelligence a question of the left side of the brain or of the right side of the brain? What can we say about that? Well, we take another step in our trip, and now we ask the next question. Well, general intelligence is great, fluid intelligence is very interesting, but what happens if we try to find the places in the brain that are relevant for intelligence and other relevant factors in the cognitive domain? So we try to analyze the commonality for fluid intelligence, crystallized intelligence, spatial intelligence, working memory capacity, attention, and processing speed. And we found an interesting thing, that again, the commonality for these 6 factors, but for some of them, it's mainly located in the right side of the brain. For fluid intelligence, here you have the main finding, for crystallized intelligence, and for working memory capacity. But not in the left side, in the right side of the brain. So what is happening here? Sometimes the left side, sometimes the right side. What's the proper answer to the main question? The problem, and we are moving now to the second part of my talk, is that intelligence is a moving target. It's difficult to find the main sites for the brain or intelligence in the brain because intelligence is a moving target. What this means? Well, this means that it's, as I said before, it's interesting to find commonalities across different studies, but it may be also interesting to find differences among studies. And when you look deep into the main evidence that was considered by the PFIT model, you see that there are many differences between the different studies considered. So I showing now this picture that shows that there are only 2 main areas in the brain that reach 50% of agreement across studies. So there are much more discrepancies than commonalities across the different studies, and this perhaps is telling something. This is not bad, but perhaps this is telling something about the main goal of trying to find the brain support for intelligence. And for trying to find some more information about that, we designed a study using 3 different samples from 3 different labs in 3 different countries, and trying to control for things that may explain why are there differences among the different studies. So what we did here was just studying general intelligence, but use samples that are matched in the G-scores with respect to sex, age, and handedness. And to give an example, if you are using a right-handed 18 years old male with a G-score of 90 in sample 1, you will have the same in sample 2. And this for the different subjects in both samples. We are trying to match both samples in those 3 places. And we ask the question, if we are matching those samples, do you think it will be possible to find the same brain correlates for these 2 samples? Okay? Well, I am presenting now just one example, and I am focusing in cortical thickness, just in cortical thickness. Okay? 3 samples. We try to find the cortical thickness correlates of differences in the general factor of intelligence in matched samples. Okay? Well, Miguel Burgareta, that is here, is responsible for this analysis, so if you find something odd here, it's his fault, okay? Well, I will present two main findings about this comparison. The first one is for the whole group in these 3 labs. The first one is from our university in Madrid, the second one is from New Mexico, and the third one is from Canada. And the 3 samples, we extract the G factor score for the 3 samples and correlate the differences in G with the differences in cortical thickness. Across these 3 samples, you see that there is no any agreement. So, what is intelligence in the brain? We don't know. Is that relevant? We'll see. Okay? There is no any similarity. There are very small similarities across different samples. And now we move one step further, and now we ask the next question. What happens if you are using the same sample, split the sample, match the subjects in both samples? Okay? So sample 1 and sample 2 are very similar in many aspects. And try to answer the question about the cortical thickness correlates in both samples. What we can see here, there is no any similarity at all. Very small similarities for the same lab using separation in 2 samples. So you see my point here. The question is, what in the brain is intelligence? We don't know. Is this relevant? We will see. Okay? Well, as my friend and colleague Rich Heyer says, perhaps the main message from this type of findings is that we must assume that not all brains work the same way. Perhaps we are trying to answer the wrong question. And this is what my colleague Kenia Martinez, that is also here, so anything about that, ask her, okay, is trying to answer now. He's just finishing a pretty exciting research trying to answer this question, where in the brain is intelligence? And she did that using 3 different imaging protocols, using different gray matter factors, and using equivalent samples. And I am not explaining what she found, because it will be much more interesting for her to explain that than for me. But I will show just one thing that it's pretty troubling. Just one thing. What happens if you compare these 3 imaging protocols for analyzing the same brains, okay, the same brains, and you compare what happens in these different protocol, imaging protocols, and compare with what we know about the brain in post-mortem studies, okay? We analyze the cortical thickness distribution across the brain that we obtain by 3 different imaging protocols in different labs. And you see that there is a very low overlap across imaging protocols for analyzing the same brains. And of course, there is a very low commonality with what we know about cortical thickness distribution from postmortem studies. Okay, so if you cannot find commonalities across imaging protocols for analyzing, just for instance, cortical thickness, it's very difficult to find commonalities across studies when you correlate cortical thickness with intelligence. Okay? It's the expected finding. Well, another important message from this analysis is that there is one of the three imaging protocols. It's pretty similar to the postmortem findings. And the troubling thing about that is that this one is the most similar to the postmortem findings, requires a lot of manual work. So doing things, pressing a key is exciting, but dangerous. So if you are interested in obtaining reliable information about, say, cortical thickness, do the hard work and work with your hands, not just press the key. Okay? Well, this is also with other studies that analyze an interesting topic that is functional connectivity. When researchers analyze functional connectivity, they reach the conclusion that you can see at the bottom of this slide, that the fact that people think or behave differently from one another is rooted in individual differences in brain anatomy and connectivity. Regions predicting individual differences in cognitive domains are located in regions of high connectivity variability. So this variability is there, and I think it's very important to pay a close attention to this fact, working with scientists from other areas for answering the question that we are interested in, that is intelligence differences. You see here the distribution of variability in this connectivity index, and this connectivity index, some people think, is very important for defining individuality. Perhaps you know something about connectomics, and some think that these connectomes for each individual are important for defining this individuality. So there is a lot of variation in these functional networks, in the same way that there are variations in the connections in structure, in brain structure. So just a brief example of why I am saying that there is a great variability, that there is a lot of change, that is very difficult, and maybe it doesn't make any sense to try to find the relevant places in the brain that are important for understanding differences in intelligence. And I will show you just a brief example, and we are reaching the final part of my talk, that how a very simple practice with a commercial video game can change brain connectivity very easily. Okay? So this is published in Human Brain Mapping, and we here analyze two groups, the standard design, one group playing the video game, and a control group. And we analyze several networks in the brain that took the information from a resting state paradigm. Okay? We analyze the connectivity among different regions in the brain using identify networks. And we analyze if there is changes in these connectivity patterns from the pre-test to the post-test in two scans, and we found that there is significant changes in these connectivity patterns that I will show you in a brief animation. okay? And you will see how the different areas of the brain that define the networks change from the before playing and after playing, comparing the control and the training group. Okay, so there is a change in connectivity and just playing a game. So we can think that other types of activities can change many different things. So this is highly dynamic. So it doesn't make much sense to answer the question of where in the brain is intelligence, but perhaps we now understand why this question is not as particularly relevant for that. Okay, arriving now. Okay, we are reaching the final stage of my talk. And let me say a couple of things. For improving our knowledge about how the brain is related with intelligence, how the brain supports intelligence, how the brain is behind intelligent behavior, we need to cooperate with scientists from other areas. These scientists is now doing a very good work, and they need more psychologists, that, for instance, the people here in this room, that must begin to work with these guys for trying to increase our knowledge about how intelligence is based in the brain. One of these projects is the Human Brain Project, that when you read the main report that supports this project, they speak about that. They try to find, to build a brain-like intelligence. They are looking for that, but there is no any single researcher from the Issid, for instance, of the ISIR there. No one. But they are trying to find that. So I think we must be involved in this kind of big projects. This is one of the, but as you probably know, Americans are also doing things about that. They are now beginning with the Brain Activity Map project that is trying to do something different but closely related. They are trying to find how the brain works, but they are not trying to build a robot or something like that. They are trying to put something inside the brain and analyze how the brain works. And the main idea of this BAM project was this guy, and this guy was from my university. That is great to tell you that Rafael Juste was from Spain and now is at the States trying to push this interesting project, okay, the BAM project. I like this one. Well, so at the end of the day, at the end of my talk, the main question is, can we do this? And let me show you, just for 5 minutes more, let me show you another video, okay?
Mr. Speaker, the President of the United States.
If I could fly like birds on high, then straight to Orion I'd go sailing.
Mr. Webb, Dr. Gilruth is here.
Bob, he's just getting into it now.
Good. Can you turn it up, please, Jim?
Their large rocket engines, which gives them many months of lead time, and recognizing the likelihood that they will exploit this lead for some time to come, still more impressive successes, we nevertheless are required to make new efforts on our own. For while we cannot guarantee that we shall one day be first, we can guarantee that any failure to make this effort will make us lost. I therefore ask the Congress, above and beyond the increases I have earlier requested for space activities, to provide the funds which are needed to meet the following national goals. First, I believe that this nation should commit itself to achieving the goal, before this decade is out, of landing a man on the moon and returning him safely to the Earth. No single space project in this period will be more impressive to mankind or more important for the long-range exploration of space, and none will be so difficult or expensive to accomplish.
Whoa, Buster.
I sure didn't waste any time. If we had gotten shepherded to space before Gagarin, it'd be over. We'd have beaten him, that would have been that. We wouldn't be talking about going to the moon for another 20 years. Between this and the Bay of Pigs. Does anybody want my job?
That 5-page memo from LBJ, pressing us to do it by 1967.
Before we get anywhere near the moon, we'll need to put thousands of man-hours into space. So far, we got 15 minutes and 22 seconds. I'm serious now. Who here wants my job? Bob, can we do this?
We'll need thousands of people, special facilities, technology and material that haven't been invented yet.
Yeah. Can we do it?
Put a man on the moon in 9 years. Yes. Absolutely. We have to.
So we can go from the Earth to the brain and have some good findings there. But for doing that, you must accept my main message that we must begin for something. And the best bet for doing something interesting is focusing on intelligence. There are many interesting traits there, but it's a good bet to begin with intelligence. So let's begin as soon as possible. Thank you very much.